Hi, my name is Morgan and I'm a neuroscience researcher, and welcome to Neuro 101, a new series I'm starting. If you're the type of person who hates long intros and like listening to the backstory and the reasoning behind the video, you can skip to this timestamp. For everyone else, I want to talk a little bit about what inspired this series. So several months ago, I got an email from someone asking me if I had any recommendations for learning the basics of neuroscience. And for anyone watching who might be wondering the same thing, I do really recommend the channel 2 Minute Neuroscience. I will put a link to them in the description. But as much as I love them and admire what they do, I don't think it's really a great place for beginners to start, because you sort of have to know what you're searching for in order to learn it, if that makes sense. So I didn't really have any great recommendations for that person, and I actually never responded to their email. So if you're seeing this, I am so sorry, but you did inspire this whole series. When I got that email, and honestly for the last several months, I was going through a pretty extreme burnout period. So burnout is something that a lot of people experience in all sorts of different industries. For people who work in research facilities like myself, or who work for universities, it's pretty common to experience a lot of burnout because of sort of the way that we basically work ourselves to the bones, then go through a period of burnout, take a little bit of time for self-care, and then restart the whole cycle. That sort of behavior is very normalized in research, which I don't think is a great thing, but it is the way that it is. So while I was going through this burnout period, I was looking for some ways to sort of spark some joy, because I love neuroscience. I love learning about the brain, and really my frustrations and my burnout is with the industry that I'm in, and with how research is conducted, and with how universities conduct research, and not with the learning about the brain and with neuroscience itself. But I was starting to project those feelings onto neuroscience, and even just thinking about brains just made me really upset. So I kept on thinking back to that original email, and one thing that I am extremely passionate about, and one of the reasons why I started this channel, is teaching people about neuroscience. And so I decided, you know what, maybe this will help me get out of my funk. I can be the resource for people to learn very basic beginner neuroscience. And throughout the process of researching this series, it really did help me regain my love for neuroscience, because the stuff I'm going to be going over in this Neuro 101 series is all of the stuff that initially made me fall in love with the brain, and all of the stuff that I love. Very basic science, very basic neuroscience is truly what I'm passionate about, and learning the in-depth inner workings of our brain. So I'm going to be teaching this Neuro 101 series as if I were teaching a class of people who may or may not have had high school biology. You do not need to come to this with any knowledge about the brain. All you need to have is some curiosity. These videos will build upon each other. So if you're watching one of my videos later in the series and you're that word is not very basic, that word doesn't make any sense to me, it's probably because I explained it in a previous video. If there's something that I talk about and you'd like to learn more about it, I will oh, my cat is in the shot. If there's something that I talk about and you would like to learn more about it, and you'd like to go more in depth, I will try to include links for more information on each of the topics in the video description. Also, throughout this series, I'm going to be using a variety of resources, like my handy dandy whiteboard, which I'll draw stuff on. I have some little plushies that I'll use to show things. These are from Giant Microbes. This is not a sponsorship. I genuinely love Giant Microbes, and if you have any scientists in your life, or if you're interested in science, I highly recommend them. They have plushies for just about anything you can think of. And then I'll also have some homemade stuff. Sorry, he has something rattling around in his brain, I guess. But I made this out of a Halloween skull. So if you see something, you're wondering where I got it, either I made it or it's from Giant Microbes. Last thing I wanted to mention, my goal is to post these videos weekly. Right now I have 10 videos planned, so hopefully I'll post one every week for the next 10 weeks. And then in the future, just as I come up with stuff that I'd like to talk about. But if you have been following this channel for any period of time, you know that I'm not great with posting schedules. I've tried to do that a lot of times in the past. So once a week is my goal, but I make no promises. So now that the intro's out of the way, let's get on to Neuro 101. For this episode of Neuro 101, we're going to be talking about how our neurons communicate with each other. So when we think about a brain, we think of a solid object, something that you can hold and you can see, you can look at it. And that is true. Our brain is something that you could pull out of the skull and hold. But every brain is made up of a bunch of these guys, like billions of neurons. As I mentioned in my first Neuro 101 video, there are two different types of cells that are in our brain. There are glia cells and there are neurons. Today we're going to be mostly focusing on neurons. So first thing, let's do a quick refresher on neuron anatomy, or like the different parts of a neuron. Over here, this is called the soma, or the cell body. Sorry, my handwriting is terrible. This guy is called the axon. And these guys over here are all called dendrites. So there are a bunch of different types of neurons and they can all look a little bit different. But this is sort of the main structure of a neuron and how we can sort of simplify it to make it easier to talk about. The general flow of information is that these dendrites are going to like pick up some information. Something else, probably another neuron, is going to communicate something to them. They're going to take it over here to the cell body. Then at the cell body, there's this spot right here that's called the axon hillock. And if the cell body decides that this is information that needs to be communicated to another neuron, it's going to send that information over to the axon hillock, and the axon hillock is going to send it all the way down to the end of the axon over here. And this spot right here is called a synapse. But this is just one half of a synapse. So now I'm going to draw a different picture. I'm just going to zoom in on what's happening right here. So this is what's called a synapse, and it's where two neurons communicate. So we're going to pretend like this guy on this side is that neuron that we were initially looking at. We're going to call it neuron A, and this is a new neuron. It's neuron B. So based on the flow of information that I was telling you about a second ago, the information is flowing down this axon over to this first half of the synapse. So let's ignore this neuron for now. We have this first half of a synapse, and this is what's called the synaptic cleft. And everything that's happening on this side is called the presynapse. And everything that's happening on this side is called the postsynapse. And again, this is all based on the flow of information. So information is coming down this axon into the synaptic cleft. Some stuff is going to happen over here that we're going to talk about. And then it's going to go into this new neuron. And I just want to acknowledge really fast, I know this is already getting a little bit complicated, especially for Neuro 101. In my personal opinion, this video is probably going to be the most complex out of all of the Neuro 101 videos I'm going to make, but it's also really important for understanding how our brain works. The point of me saying that is, don't feel bad if you don't understand this the first time, or if you have to look up multiple videos. Sometimes the way that one person explains it isn't the way that's going to make sense in your head, and that's okay. So just to zoom out a bit of what I just said, I'm going to use these two guys. These are two different types of neurons. And just to orient us again, this guy is the cell body, the axon, and the dendrites. This one, I know he looks a little bit different, but up here, this sort of like ball-ish shape is the cell body. These guys up here are the dendrites, and then this long bit is the axon. So we're going to say this guy is neuron A. So neuron A is going to get some information from its dendrites. It's going to go into the cell body and down through the axon. Now over here at the axon, that's going to be near this guy's dendrites. And they're not actually touching, they're just next to each other. And some information is going to go from this axon to this dendrite. And then same as earlier, from the dendrite down into the cell body, down the axon, onto the next one. So back in ye olden days, when we couldn't super zoom in on what is happening in the nervous system, we actually used to think that neurons were touching each other. So instead of this space right here, these neurons would just be like right next to each other, and they would just be sending these electrical signals back and forth. But then later experiments came along and people figured out actually there is a little bit of space here. So the neurons aren't actually touching each other. That's why it's important that we acknowledge that even though we think of the brain as one solid thing, these neurons are not physically touching. The space between them is so small that you could basically pretend like it doesn't exist, but it does exist. So that's the really fast, oversimplified version of how neurons talk to each other. Now we're going to get into the more nitty-gritty. So the first step to understanding how the brain communicates is understanding how it generates its electricity, or how electricity works in the brain. And this sounds scary, but I'm going to do my best to make it as simple as possible. So here at the bottom, I've drawn a neuron, and then what I've done here is I've drawn a circle to represent the neuron. And all that is it just makes us easier to think about than if I were drawing stuff along this entire thing. We're just going to pretend like it's a circle. Now, the very basics of cell biology is that cells are surrounded by a membrane, and that's what keeps like the outside of the cell outside and the inside of the cell inside. That membrane is made up of the phospholipid bilayer. The phospholipid bilayer is like its whole own thing. You're more than welcome to look up videos on it. But the main point of me talking about it now is it's made up of these little guys. I know this is going to look a little weird at first, but this is how people draw them to represent. It's two different layers. So we have this bit that's on, this is the outside of the cell, and this is the inside of the cell. These little guys are called phospholipids. There's two layers, phospholipid bilayer. The important thing to take away from this, and honestly the important thing to keep in mind throughout all of neuroscience, is even though things look solid, if you zoom in, everything is made up of smaller parts. So even though this membrane looks solid to us, it has these little parts in it. And the phospholipid bilayer, even though it has like these little separations, does a really good job of keeping things outside of the cell that need to stay outside and things inside the cell that need to stay inside. But because they have these spaces between them, what we can do is we can come in and insert little extra holes called channels. Or there are some other things called pumps. They're similar concepts. The difference between a channel and a pump is that a channel doesn't take much energy and a pump does take some energy. So even though for the most part this membrane looks solid, it has inserted into the membrane some channels that can let things in or out depending on what we need. And channels can be used for different stuff. In neuroscience, our main concern is ion channels. So even though the cell, the neuron, is made up of a bunch of different things, there's stuff going in and out, there's a lot going on in one single cell, our main concern here are ions. These ions are moving in and out of the cell super duper fast. So some of the channels are going in, some of the channels are going out. About 100 million ions can move through a single channel in a second. So there are a bunch of different types of ions. In neuroscience, we're mostly concerned about sodium, potassium, and calcium. And if you notice these little plus signs that I put up there, that's because every ion has its own electrical charge. So if you think of these ions that have their own electrical charge moving back and forth across the membrane super duper fast, that's going to also generate an electrical charge. We refer to this charge that these ions have generated as an electrical gradient or a concentration gradient. So just to do a bit of an example, because I know this is really complicated, let's say that potassium is moving in and out of the cell. And because potassium is moving in and out of this channel, we wind up with 10 potassium on the inside of the cell and let's say 5 potassium outside of the cell. So we would say that there's a positive gradient inside the cell because there's more positive charge inside the cell than there is outside the cell. And anytime we talk about these gradients, we're comparing what's happening inside the cell to what's happening outside the cell. So through knowing the charge of these ions that are going in and out of the cell and the electric charge that they're building up by moving across this membrane, we can figure out the electrical charge of the neuron. And to do that, we use a lot of math and equations that I'm not going to go through now because that's a bit too much for a Neuro 101 video, but just know that there are ways that we can determine the charge of the neuron. Something to mention real quick about these channels is that there are several different types of channels, and channels normally only let through like 1 or 2 types of ions. So this guy might be a potassium channel, and maybe this one up here is a sodium channel. And they can have different requirements to let things in or out. One of the most common types of channels we deal with in neuroscience is what's called a voltage-gated channel. And that just means that the cell has to have a certain voltage in order for that channel to let things in or out. Another common type of channel is a ligand-gated channel, and that one's a little bit harder to explain just because ligand is a weird word. But basically what it means is this is a channel, things can go in or out, and it has this spot here that it needs something to bind to it in order for it to be open or closed. And the thing that binds to it is called a ligand. So let's say we have a neurotransmitter out here. It's this nice circle shape. There's a spot right here for a circle to bind. So it comes and binds here, and that's going to determine whether this channel is open or closed. Last but not least, there are channels called leaky channels. And that name makes them sound very unimportant, but they actually are incredibly important for maintaining the cell. So leaky channels are named that way because they're open all the time. And the reason why they're so important is because of things like the sodium-potassium pump. So the sodium-potassium pump is really important when it comes to generating action potentials, which we'll talk about in a minute. But essentially what it does, it takes 3 sodium and moves them out of the cell, and then 2 potassium and moves them into the cell. And if that were the only thing at work here, eventually this cell would run out of sodium because all of the sodium is getting pumped out. But sodium is really important to the way that the cell functions, and it's really important that the sodium-potassium pump has sodium that it can keep on pumping out. So up here is a leaky channel where sodium is constantly coming in. So even though it's being pumped out, it's also being let in by these leaky channels. So leaky channels are just really important for maintaining the balance of the cell. Now that we've talked about how ions can move across the membranes and generate an electrical charge, we can finally talk about how neurons use electricity to communicate. Neurons use action potentials to communicate. An action potential is just an electrical signal that we're using to send information. I am going to warn you now, I'm about to throw some numbers at you. Just know, like I said earlier, we're able to calculate these using equations, looking at the different ions and how they're flowing back and forth. But the important thing is just to know that we are able to get these numbers from something. I'm not just pulling them out of my head. So your average neuron is going to be sitting at around -70 millivolts. That's its normal charge when absolutely nothing is happening. So if nothing is going on in the cell, there's no ions doing anything that they're not supposed to be doing, there will still be a flow of ions back and forth, but they're just sticking around at this -70 millivolts. So it's just going to be a flat line looking like that. Then let's say something happens to make the neuron want to communicate. So a neurotransmitter comes along, the ions start flowing in a different way. Something happens to where the neuron is like, I need to send some information out. So it's going to start generating an action potential where it starts to go up like that. And the way it does that is by letting a lot of sodium into the cell. And that's going to send the action potential up like that. This is a process called depolarization. So if a neuron is hanging out at this -70 situation, we say that the neuron is polarized. Now it's being depolarized, so it's going up. And then to stop firing, the action potential starts to go down. And this is when a lot of potassium is being let out of the cell. And this is a process called repolarization. And it's just going to go back down to its normal resting state. But a lot of times it will have this moment of sort of overcorrection where it actually goes below its normal resting state. And that is hyperpolarization. And then it'll come back up to its resting state and go about its day. So this is how the flow of ions in and out of the cell can generate an action potential, which is the neuron's way of communicating. But not every time that sodium starts going into a cell is an action potential going to happen. These neurons have what's called a threshold. That's not a good line. And a threshold is sort of the electrical point that the neuron needs to reach in order for this action potential to happen. If something happens to where the neuron is sort of starting to depolarize, but it doesn't meet that threshold, it's just going to go back down and nothing is actually going to come of it. I'm not going to lie to you, action potentials are kind of tricky, and I still have to look up whether it's sodium going in or out and potassium going in or out whenever I talk about this. The most important thing to remember from this is that the flow of these ions going in and out of the cell generates an electrical charge that can go above the threshold of the neuron to cause it to fire. People might also refer to this as spiking, because whenever these guys happen really close to each other, what it'll look like is like boop, 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 boop. It looks like spikes. And this is super duper zoomed in onto the neuron. So let's say that this is our neuron down here. What we're saying is that right around here is what's called the axon hillock, which is where most action potentials start. So essentially the cell body decides that it needs to send an action potential. The sodium and potassium start flowing in their little ways, and an action potential starts here. It travels down the axon and gets to the synapse, where it then causes the release of neurotransmitter. And that's what we're going to talk about next. But first I have to take a little bit of a detour to talk about myelin sheaths. All right, so we have our neuron here. And we're saying that an action potential is going to travel from here all the way down to the synapse. Something that can help the action potential along is the myelin sheath. So remember how a few times I've mentioned glia cells and said that we're not really going to talk about them too much? This is one of the instances where we are going to talk about them, and they're incredibly important. There are a bunch of different types of glia cells, but the two that we care about right now are called Schwann cells and oligodendrites. Oligodendrites are my favorite just because that's a super fun name. So the oligodendrites, or the Schwann cells, are just going to be hanging out up here, and they're going to reach their little legs out to a bunch of different neurons, not just one neuron in particular. And they're going to come along and wrap around it and just form like some insulation. That's not a great picture, but you can look at better pictures. So now you have parts of the axon that are covered by the myelin sheath. You also have these little spaces here, which are called the nodes of Ranvier. The nodes of Ranvier. That's supposed to be an A. And what happens here is sort of counterintuitive, because what you'll hear a lot of people say is that the action potential will start over here and then it'll sort of jump to these nodes like this, and that helps it get to the synapse faster. But the action potential isn't doing any jumping. What's actually happening is that these nodes are sort of giving them a little speed boost. Like in a video game, whenever you like cross like a certain threshold and it makes your character like run extra fast. So because there's this extra insulation happening in these myelin sheaths, that causes action potentials to get like a boost in these nodes because there's an increased flow of sodium and potassium happening. It's really complicated to explain, but the point is that the action potential is sort of like going, speeds up, going slow, speeds up, going slow, speeds like that. And that will help it get to the synapse faster. Not all neurons have myelin sheaths, which means not all neurons have this ability to communicate a little extra fast. So some neurons have this little speed boost and some of them don't. There are also several autoimmune disorders that will eat away at the myelin sheath and sort of get rid of it, and the neuron will either go back to sort of the same like slower state of the other neurons who don't have a myelin sheath, or it can also just cause the neuron to sort of break down entirely. The most famous example of this is multiple sclerosis, but there are other autoimmune disorders that cause this to happen too. So for a quick recap, now we've talked about how a neuron at sort of like the cell body is able to send information from the cell body down to the synapse. Now, how does the neuron get that information from the synapse, the presynaptic side, over to the postsynaptic side to be able to talk to another neuron? The bit that we've talked about so far is done through electricity. Now we're going to talk about chemical communication. Something that's really important for me to mention before we get into this fully is that all neurons have this action potential communication, and then they're doing this chemical communication. So if they're all sort of talking to each other in the same way, how are they able to say different things? That's done using different types of neurotransmitters and different receptors for those neurotransmitters. And those two topics are complex enough that they're actually going to be the next two videos in this series. So for this one, I'm just going to talk about things very broadly, just saying the neurotransmitter and its receptor. And then in the next two videos, we'll get into the different types of neurotransmitters and the different types of receptors. So a neurotransmitter is just a chemical that neurons use to communicate with each other. And neurotransmitters are usually made in the cell body, and then they have to be taken down the axon. Once they're carried down the axon, then they come into this synaptic terminal, or this area is also called the synaptic button or button. The neurotransmitter is in these little pockets that are called vesicles. And so there's just some neurotransmitter chilling out in there. I'm not going to get into how neurotransmitters are made or how they're carried down the axon. Just the main point of this is that there's not like an infinite amount of neurotransmitter in the neuron. It has a certain amount that's being produced in the cell body. It has a certain amount that's in transit. And eventually there's only a certain amount that is in the synapse. So each time an action potential happens, one of these vesicles releases its neurotransmitter out into the synapse. So let's say, hypothetically, this neuron fired 5 action potentials. So now, by 4 action potentials, it's released all of its neurotransmitter. And we're saying that these happen so close together that the neurotransmitter that's traveling down the axon hasn't had enough time to get here. That would mean that the 5th action potential that happens, even though it should release neurotransmitter because action potential hypothetically does release a vesicle worth of neurotransmitter, since there are no vesicles there or no neurotransmitter to be released, nothing actually happens. So it is possible to have an action potential and have no communication that actually happens. But most of the time, an action potential will cause the release of a vesicle's worth of neurotransmitter into the synapse. So this is all great. We had managed to get an action potential from the cell body down to the synapse. We managed to get neurotransmitter from the cell body down to the synapse. And the action potential caused the neurotransmitter to be released into this space in the synapse. So neuron A over here is done. It's done its job. It's sent the communication out into the world. But just because neuron A has released all of this neurotransmitter doesn't mean that neuron B is going to pick it up. So how does this neuron pick up the communication that was sent? It does that through things called receptors. So like I said, receptors are going to be their own thing in their own video because they can be incredibly complicated. But the point is, if this neuron releases some neurotransmitter, nothing is going to happen unless this neuron has a receptor that's ready to receive the neurotransmitter. Depending on the type of neurotransmitter that's released and the type of receptor that it's binding to, a certain amount of neurotransmitter will bind to a certain amount of receptors up here, and then that will be enough to generate an action potential, a new action potential that will travel down this neuron to its cell body up here. So I know this video has likely been incredibly complex and a lot to take in. Again, do not feel bad if you have to watch several different videos to be able to put all of this together, or if you need to rewatch this one, or if you still have questions. That is perfectly fine. This channel is all about learning. I'm gonna do my best to try to recap what we've talked about so far. So we're saying that neuron A got some information. So it got some info that went down the dendrites, went to the cell body. Cell body thought about that for a minute, was like, do I need to send this out? And decide, yes, I need to communicate this to another neuron. So it goes over to the axon hillock. And is like, hey, go tell neuron B this information. So over here, there's an exchange of sodium and potassium and other ions that will generate an action potential. Then the action potential travels down the axon, and it may or may not be aided by myelin sheaths. Once it reaches the synapse, it's going to cause the release of neurotransmitter. And then if neuron B has the right receptors for that neurotransmitter, this is going to send a new action potential to this cell body. This cell body is going to think about it for a minute, decide whether or not it wants to send this information to someone else, and then possibly send another action potential down to its axon, and then the process just repeats. So the very last thing I want to talk about in this video is how do we stop this process? It's great that we've done all this communicating, but once we've communicated what we need to say, we shouldn't continue to release neurotransmitter here. We don't need to keep on sending these action potentials. We need to stop at some point. There's a few different ways that our neurons do this, and it usually involves a feedback loop. So the first version of a feedback loop would involve another neuron. So let's say after neuron B, there's this other neuron here. It's going to pick up some of this information, and its axon is going to go all the way over to neuron A, and neuron A has a dendrite here that's going to pick up this information. So what'll happen is neuron A communicates its information to neuron B. Neuron B comes and tells neuron C, hey, this is the information that I've received. Neuron C is going to come and tell neuron A, hey, don't worry, neuron B got the info, we're all good. And then neuron A will stop sending the communication. Usually these loops are a little bit more complicated, but we don't have to get into that. Another way would be if neuron A had its own receptors over here. So not only is the neurotransmitter that neuron A is releasing coming to neuron B, but a little bit of that neurotransmitter is coming back to neuron A and binding to these receptors. And that is going to send a message back to neuron A that says, hey, we've released the neurotransmitter, we've released a lot of it, we're probably good. And those are sort of the two main ways that your neuron knows that it can stop firing. We also have to deal with all of this neurotransmitter that's being released because not all of it is going to come and bind to neuron B. Some of it's just going to hang out and it's going to be released in your brain just chilling, and it has to go somewhere. So one way it can do this, again, is that neuron A will have some receptors for it, and neuron A will sort of like suck it back into itself and recycle that neurotransmitter and reuse it again later. And this is a process called endocytosis. And then another really interesting thing is that those glial cells that I've mentioned a couple of times might also be over here, and it will just sort of vacuum up the extra neurotransmitter. So I hope this video has made sense. I hope that you've learned a little bit more about how our brain is able to communicate and how our neurons communicate with each other. Like I said, I know it's a bit much. I'm going to include in the description a few other videos that explain how our brain communicates, that might explain it differently than how I did, in case you might want to use that as a resource. And I'll also include some videos that go a little bit further in depth than I did, if that's something you're interested in. Like I've teased a few times now, the next 2 videos in the series is going to be one on neurotransmitters and one on receptors, to talk a little bit more about the specifics of if this neurotransmitter is released versus this neurotransmitter, what will happen, how are neurons able to communicate different things when they're all using the same method of communication. But that's all for this Neuro 101 video. I will see you next time. Bye.